webinar that we have coming up in September is with John Watts. So he's a great guy to have conversations with and to learn things from. Very willing to chat about his career trajectory, his research. So you won't want to miss that session. And then we have a few more researchers leading up to the end of the year. But remember, we do have our annual meeting. So around the time of the annual meeting, we'll slow down a little bit on the webinars, and then we'll pick back up for the end of the year. So I'm just going to pass it on to Dan to get started and to introduce himself a little bit more as he's kind of giving his presentation. So he will take questions at the end. If you have questions throughout, though, feel free to put them in the chat or in the Q&A function, and then we will moderate them for him at the end of his talk. All right, Dan, take it away.
Thank you very much, Erin. Let me just figure out how to work the Zoom. So hopefully everyone can see my presentation and not presenter mode.
Yeah, we can see it properly.
Okay, good. Thank you. Okay, so yes, hi. For those of you that don't know me, my name's Dan O'Reilly. I've been as Erin mentioned, I've been a trainee rep on the OTS Board of Directors for the last few years. I'm also a third-year postdoc in the Correlate lab now. How time has flown. And I primarily work on Huntington's disease, and I also have a lot of patient advocacy that I do. Just to give everyone a brief background on Huntington's disease, it's a rare neurodegenerative disease that's caused by an expansion of the CAG repeat tract in the Huntington gene. Basically, if you have less than 40 repeats, you will not have Huntington's disease. But if you have anything greater than 40, you will get Huntington's disease. And what happens in the brain is that from these patient MRI scans, you can see on the left that over time, patients get, basically their ventricles will degrade to get larger and larger, and essentially they are degrading, and this leads to symptoms such as chorea, which is shaking, a lot of motor dysfunction, cognitive dysfunction. Now, something that I have a lot of knowledge on Huntington's disease because my mum had Huntington's disease. You can see from the pictures of the right. This is my mum. On the left is my mum before she had HD, and on the right is a picture of her with HD. You can see that she's lost a lot of weight. What's not shown in the pictures is the chorea and the impact it has having a loved one who has a disease on her and everyone around her. But what this did inspire was for me to get into patient advocacy. And what I do know is that anyone who has been in patient advocacy or has been around someone that has a disease really, really wants to help out getting a therapeutic for that disease. Like, that is that they set that as their goal in life, and that's kind of what I have done. Now, in realistic terms, we know the drug development pipeline, it is a long haul. A lot of things fail in preclinical stage, and even if things are successful, it can take a decade to get something approved by the FDA. And really, it takes many, many people. It takes people from industry, it takes people from academia, and it's also important to talk about the patient's role in this, because they actually can play patient and advocates can play a very key role in getting a drug from even preclinical research to clinical and beyond. Now, what roles can patients and advocates play? And when I talk about advocates, I mean anyone that actually is involved or has been around a patient, carer, family member, that type of thing. Now, advocates and patients can take part in clinical trials, natural history studies. They can help design clinical endpoints, which is very important for studies because they can tell you exactly which part of the disease matters most to them, what affects them on a day-to-day basis, not just what you read in the text might be very, very different from actually what they care about on a day-to-day basis, which can help you design your clinical studies better. They can also educate researchers about the disease. And actually this can go both ways, because if you have patients and advocates involved in research from an early stage, you can educate them about certain aspects of preclinical research or anything that you're doing. So this is really a two-way street. And then the final two things go hand in hand, which is raising awareness and funding. Now, the patient community, the patient and advocate community is very well together, and it's a great community. It's like the OTS community. Everyone is willing to help out everyone, and raising awareness through either doing walks, all these kind of events that they do really can help you as a researcher to also then get funding as well, because they can raise money through donations, or maybe they have can get bigger donors, or so there are things that real patients can actually impact preclinical research. So I think the biggest example of this recently has been with Milasen, the first NF1 drug, where the mother of Mila played a pivotal role in getting her a diagnosis and making sure that she got this drug that was developed for her. And now, since Milasen, there are loads of researchers out there that really want to work on NF1 drugs and are trying to interact with patients more on a day-to-day basis at an earlier point in the development process for whatever disease that they're going after. Now, it is important when you're talking to patients, the communication between patients and yourselves as researchers needs to be very good. A lot of times when researchers talk about their research, they can kind of put more of a positive spin on everything. And I think when you talk about with patients, they just want an honest conversation. You know, setting realistic expectations within yourself and for the patient, so that it might take us a few years, but we are going to try and get a disease, is better than saying we will get there. There'll be a disease modifying drug in 2 years, which is unrealistic at best. The other thing I would highly recommend is, if you're going to talk to patients, is also discuss negative results, because actually negative results can have a big impact on research going forward. And whilst you might think that the patient doesn't want to hear about negative results, they're interested. They're happy to hear about everything, even the negative results. And so during the rest of my talk, where I talk more about research, I'll also highlight how patients have been involved in Huntington's disease that have led to a significant move forward in our understanding of the disease progression. So that takes me back to HD, where, as I said, in Huntington's disease, greater than 40 repeats, you produce this transcript and protein that is elongated, has an elongated CAG tract, and this leads to disease. Now, for a while, the dogma in the HD field was that if you silence the mutant gene, that should stop the disease progression. And actually, there was an ASO that was in clinic by Roche and Ionis that did that, silence the disease, the mutant Huntington, and was hoping to stop the disease. Preclinical data was very exciting, and it got to phase 3 clinical studies, but unfortunately, it then failed. There's been a few failures for HD this year, but this is the one I really want to highlight because I wanted to go into more detail about it. So there are 3 things to discuss when it comes to this trial. The first is that at the highest doses of the study, the patients got worse, and that's probably because of tox associated with the ASO that was making their disease, basically probably the degradation got worse, and so made the disease progression quicker. The drug itself also silenced the wild type gene. Now, again, preclinical data suggested that in the mice, at least, suggested silencing wild type Huntington up to 75% was fine, had no adverse effects, but perhaps that's not the case in humans. And really the third kind of discussion point from the trial was, does silencing mutant Huntington actually impact disease? We don't know the answer to this last one, because in the like lowest dose or the middle dose, they only silenced mutant Huntington about 40%. It wasn't that much silencing, but it's still an active question. Do we actually understand the disease biology enough to say that mutant Huntington silencing will impact disease progression? And this is where my research kind of comes in. So a recent genome-wide association study found that actually the number of CAG repeats that you have, that you're born with, defines your age of onset, i.e. someone who has 40 repeats will have a later age of onset than someone who has 50 repeats, for example. And they also linked something called somatic expansion to this process, where actually what happens is you may start out with 40 repeats, but over time, that number increases in different regions, in different neurons of the brain, and that is what drives disease progression. They also found many modifiers of this disease, somatic expansion. So here's where I just want to give a shout out to patients, because a while ago, the Cure Huntington's Disease Initiative set up this Enroll HD, where they tracked patients. They take blood samples from you, and they also have you do some clinical tests and evaluate you on a yearly basis. And it was thanks to this Enroll study where they took all the patient data, and this is where they were did this genome-wide associations study from. So without the patients to go every year and to get the blood withdrawn and to contribute in that way, we'd be further behind as a field as HD. Now, this leads me to MSH3, mut-S homolog 3 gene. Basically, it was found that this gene, SNPs in the gene associated with decreased age of onset. So basically, SNPs made the disease progression worse. Mutations in the gene made the progression worse. The MSH3 gene's primary role is in the mismatch repair pathway and fixing large loops. Now, you may be thinking, well, hang on, you're talking about mismatch repair pathway. Isn't that really associated with cancers? And yes, a lot of them are associated with cancers, but knockout of MSH3 has been shown to be basically the same as, have the same lifespan as wild type. And in terms of genomic instability, it was basically the same. It had no effect on genomic instability over the lifetime of the mouse. So one question we're interested in is, can MSH3 silencing be a new therapeutic paradigm for HD? And can it block expansion of that HD gene? So this leads me to the divalent siRNA that has been previously published by the Correlate lab and allows broad distribution in the mouse and great uptake in the mouse and non-human primate brain. Basically, the siRNA are linked through the 3' ends, and this increases the size of the siRNA and prevents it from being cleared by the CNS. So here I'm showing some. You can see that the siRNA increases the accumulation in many regions of the brain, and also gives potent silencing at 1 month and up to 6 months duration of effect of the HTT mRNA expression, and also the same at the protein level. So this is really a great tool for us to use to silence MSH3 mouse in the mouse brain, and then hopefully see an effect on blocking expansion. So what we wanted to do was to take that siRNA, change the scaffold, and screen for more targeting MSH3, and does that block somatic repeat expansion? Right. First, we screened for many SIR targeting MSH3 in HeLa cells and did some dose responses to confirm that we had identified several potent compounds, some with cross homology as well. But next we wanted to actually say, well, okay, we have these compounds. Do these work in the mice? And for this, we had to turn to a mouse model of HD. This is the Q111 mice. Now, for all intensive purposes, it has 111 repeats. It has been shown to undergo expansion of those repeats, but it's in the mouse. It has a humanized exon 1 in the mouse context. So basically, it's mostly mouse Huntington gene and a small bit of humanized Huntington. And what we did was we did 5 nanomoles per ventricle injection, PBS, NTC, siMSH3 1000, which is the one that's targeting the MSH3. And then we also injected this siHTT, which has been shown to silence the Huntington, but effectively acts as a secondary non-targeting control because it doesn't block expansion. And we just confirmed that our injections work using this. And our readouts were mRNA abundance, protein levels using Western blot, and the actual fragmentation analysis to show that can we block expansion. We injected the mice at 12 weeks and then waited 2 months to take them down. So I have to give a bit of background on RNA scope, which is a single molecule RNA fluorescence in situ hybridization assay, which allows you to visualize mRNA foci. And you can see from the slide that I have, the DAPI is in blue, that's staining the nucleus. And then you can see different mRNA are stained in the different colors. And you can see that it allows us to show which is the cytoplasmic mRNA versus which is the nuclear mRNA. And you can quantitate, you can get single cell quantitations from this. So it's a very powerful tool. So we looked at the localization of the MSH3 mRNA, and we can see that it's primarily nuclear localized with 60%, 66% localized to the nucleus. You can see in more detail here that mostly the foci are in the nucleus, whereas only a small amount are in the cytoplasm. And we can quantitate the silencing based on the amount in the foci. And so we can see that once in the cytoplasmic foci, we see when treatment of siMSH3 1000, we see robust silencing of the number of foci per cell, which is really exciting. But naturally, we really cared about the protein expression because that is going to be what helps block somatic expansion if we can silence that. So here's the MSH3 protein expression. We can see that in the striatum, cortex, and thalamus, we get 75 to 80% silencing of the MSH3 protein, which is really exciting. And we also confirmed that our HTT control also silenced HTT, which confirms basically that our injections all worked well. Now, the big question is, we have such great silencing of the MSH3 protein, do we block somatic expansion? And the fragmentation analysis, basically, to do this, it's essentially PCR. And you can see from the PBS and NTC, you get these broad distributions. And essentially, each peak is another CAG repeat that adds on over the time of the experiment. And Professor Jeff Carroll, I just want to highlight, performed all these experiments and was a great collaborator for this. Now, if we look at the distribution of both the PBS and the NTC, you can see once they start, they don't stop expanding essentially. But when we treat with our siMSH3, we see that we get complete blocking of the expansion. And even better when we compare it to the HTT, that other non-targeting control, which we know we shouldn't silence the block expansion, we see that essentially that is the same as the non-targeting control. A very good sign that we have blocked expansion. Now, this is more of a qualitative way to deal with the fragmentation analysis. You can actually quantitate it through something called an instability index. So we did that. And now on the right is the instability index, the PBS, the NTC, and the 2 siRNAs. Unfortunately, because of the NTC group is a bit variable, we couldn't get statistical significance between that and the siMSH3, but luckily we have the HTT control there, which is also a secondary and non-targeting control for us. So we do see statistically significant blockage of expansion between the siMSH3 and the HTT control that we use. So whilst preliminary data, we were very confident that we could block expansion, but naturally we wanted to repeat the study. And for that, we actually chose the secondary mouse model. So this is BAC-CAG. Essentially, it has 120 CAG repeats. It undergoes expansion, and most importantly, it has the fully humanized mutant Huntington gene. So now we can be more certain, if we see blocking of expansion, that it could impact human biology. So again, we injected PBS, NTC, the same conditions at the same ages as well. And this time when we do the protein, unfortunately, we see only 50% silencing of the MSH3 protein throughout the different brain regions. And at this point, safe to say, I was a bit shocked and was thinking, Anastasia is going to kill me. What did I do wrong? But luckily, we still did the fragmentation analysis, and we actually do see statistically significant blocking of expansion between the NTC and the siMSH3, which suggests that actually you only need 50% silencing to potentially block expansion, which is really important for particularly silencing MSH3, which is in the mismatch repair pathway. So the less you can silence it to get your effect, the better, essentially. So our next step was to do a more longer term study. This time we wanted to inject the same compounds plus an extra compound, MSH3 1468, that we had identified in the screen. But we also wanted to do 4 months duration of effect to see how long does this effect of silencing the protein last, and can we still block expansion at that time point? And we did this in the BAC-CAG mice because it has the humanized mutant Huntington gene. Now, here's the protein expression. Now we see statistically significant silencing between the MSH3 1000 and NTC, but we don't see statistically significant silencing between MSH3 1468 and the NTC. Now, when we check the fragment instability index again, do the quantitation, this time we actually see that MSH3 1000 and 1468 both block expansion, which is really interesting. And this is something that we're going to have to pursue further. The next steps in this is to actually try and define how much MSH3 silencing is required to block expansion. Is it like 20%? Is it 10%? We don't know, and clearly we're going to have to increase the power of our studies to actually investigate that, but we think it's an important point, especially when you're talking about therapeutics, and eventually you're going to want to know what dose do you want to retreat again. Well, you want to know that you're still blocking expansion even at the lower doses. So it's really exciting at the moment, and we're hoping to push this further towards more preclinical stuff and do some larger animals. So with that, I'd just like to thank everyone in the Correlate lab for all of their help and enthusiasm with this project, all the collaborators, which have been amazing, without which the data wouldn't be half as great. I also just want to thank all the funding agencies. And I just want to point out that this is the first time I'm presenting a picture of the Correlate lab where I'm in it in 2 years, thanks to COVID. So that's great. And thank you all for listening.
Thank you, Dan, for this wonderful overview of the research that you are doing, but also of highlighting the importance of patient organizations and patients' involvement in research, because I think that's an important aspect, that researchers do not only focus on their research, but that they also realize why am I doing this research and what is important about the research that I'm doing. So if you have a question for Dan, you can use the chat function or the Q&A. So I heard from Annemieke that the chat might not be working, so you can always use the Q&A. We will have a look on both of them. I will start with the first question for you, Dan. And I think we are always doing a lot of research in the lab, and I think one of the most important readout in the assays that we are using in the lab is we have a look at protein levels, or we have a look at mRNA levels, and when they go down, or we have a stability index, those kind of things. So these are things that we find important, because that means that we have an impact on the mechanism of the disease. But if we take it to the patient perspective, so what do you think is important for a patient as an outcome measure? So for an HD patient,
I mean, I think for a patient, it'll be actually having probably more a behavioral assay where you can show that the mice don't do some negative behavior that they would normally do. You know, that for them, I think that would be the most impactful. The only trouble with the particularly the HD mice is that they take ages to get disease, and even then it's not very strong. So it's difficult to do that in this case. We will do that, but it's difficult.
Then I have a question from Annemieke. So just move on the patient advocacy part. So often natural history data becomes the property of researchers or institutes. Do you think this is fair?
No, I think it should be property of the patients. And
yeah, so I think you own your own data, right? But it's difficult to own data of all the patients. So I think that's always a difficult part to.
You should own your own data, and it shouldn't be a property of someone else.
Cool. And then another question from Annemieke. So I think Huntington disease is one example of a repeat expansion disease, but there are many other repeat expansion diseases like the SCAS, but also myotonic dystrophy. So do you think that this particular approach will also help for the other diseases?
So we are looking into that. Some of the other diseases, like Friedreich's ataxia, for example, it's not a CAG repeat, but it's a GAA repeat. It might be that silencing other genes in the mismatch repair pathway would be better for those diseases, but we will still try the MSH3, and we're also working on siRNA to target other MMR genes for that purpose.
Yeah, and then a question. So I think you started your webinar with so you started with the patient advocacy, but I think in the first part was also the Ionis trial, the clinical trial. And I think if you look at the preclinical data, I think the preclinical data was really promising, right? Yeah. Amazing data in cell models, in animal models, also, and also the phase 1/2 clinical trial was really promising. But then it failed in the phase 3 trial, and then you gave a few explanations. And I think one of the explanations that you gave that it might also impact, so it might be toxicity of the ASO, so that might be one. And another explanation was that it can also have you lower the wild type level, so it might also impact the lowering of the wild type levels might also impact the patients. So the new approach that you are now using is now you are lowering the MSH3. So what effect does lowering of MSH3 have? And then also, you know, you use animal models to research it, right? So do you think you have the good models?
I think we have okay models. The best models would be something that has way better behavioral phenotype in a shorter time span, and something that's way more robust, that I know some people are trying to develop, but I don't think we have the best models. We can show we can block expansion, but from then on, it's a bit of gets a bit, you know, like a bit wishy-washy in terms of the behavioral readouts, which are important which are also important to show that you have some effect on the disease progression.
Yeah, and I think so. When I look at the questions, I think this more or less covers also the question of Alex. So maybe you can comment also a bit on this. So thank you, Dan, for the great seminar. Following Ron's question and your comment about the model, do you think that still animal models can deliver reliable data that we can extrapolate to humans, in particular for Huntington? I'm asking because in other diseases we do not have models because mice do not show the same phenotype. Could you comment on that?
I think no, they cannot. Maybe at a molecular biology level, just looking, for example, just if you want to block expansion, show that you can block expansion, that type of thing, yes, they can do it and they can be robust in that way. Anything more than that, phenotypes, no.
Okay, and question follow to this. So does the siRNA targeting MSH3 affect motor disturbances in the BAC-CAG mouse?
We haven't looked into that, but that's something we can look into, yeah. Probably should.
Okay, and then a question from Annemieke. Another question from me, why focus on siRNA rather than the RNA-H? That would also be a good approach.
Primarily because of the divalent siRNA technology, which gives such broad distribution in in mouse and non-human primate brains. That's why we took the siRNA approach, so that we could really robustly silence the MSH3 gene and get increased duration of effect.
And then a bit in I think a related question to this. So I think Annemieke mentions the RNA-H approach. I think there are several, I think there are a lot of companies working on HD and on clinical, on different approaches to treat HD. So what would be the best approach? Because, for example, you also have the approach from Wave, where they have the allele-specific approach, and they have the Unicure approach, and I think there are some companies targeting the CAG repeat. So all different approaches. So what would be the best, or do you think it's?
I think it's still up for grabs, because the problem with HD is that there is no understanding of the mechanism of actual toxicity. You know, yes, we know that you have a CAG number of greater than 40, you will get the disease, and there is, you know, degradation of the brain essentially. But the in-between steps of how that happens, it's still up for grabs. And so it's difficult for anyone to say which one will be successful, because, for example, in HD, there is aggregate formation, both protein and mRNA. And but there is data that suggests that that's a protective response and not a toxic response. So until we have that level of understanding, it's difficult for me to comment on which one would be successful. The good thing about silencing MSH3 is that you can block expansion before it starts happening. And if you get it early enough, then that should stop the disease. The problem is actually getting the FDA to let you treat patients before they have any clinical symptoms.
So when would you want to start treating patients? So when is there enough evidence to start patients before onset of disease? So there'll be like a particular biomarker present that you can start treating people, or just start treating as soon that we know that people have these particular mutations? Any ideas about this?
Probably as soon as they know, because there's more and more evidence that actually before clinical symptom onset, there is expansion going on, like potentially years before. And I don't think, but the limit of that has not been defined yet. It's kind of a new studies that have been going on. So as early as possible, I would say.
Yeah, so I'm doing all the questions, but I see a quick question for Annemieke. So do you think pre-symptomatic people would be willing to be treated?
I think yes, if they knew that yeah, I think they would.
Okay, question from Alex. So my other question was triggered by your comment about patient-researcher communication and how we should approach the explanation of the results in managing expectations. How should we develop a better communication? Should this be discussed in advance, what each of us expect? or whether they want a realistic answer. I'm asking this because I had opposite experiences of what you highlighted on the way to communicate.
I mean, I think the ways I highlighted are just some things that I have found, and everyone's going to have different experiences with that. I think when you go to patients, it's just best to chat to them about their expectations and your expectations, and learn how to explain your research in a way that they can understand and grasp, so that they have enough knowledge about the pitfalls as well. And so to be really upfront about that. Like saying the chance of, yes, we've identified this siRNA or ASO, but the chance of it actually working in a mouse model is lower than you would expect. So saying things like that is important.
This question from Feng. Great talk. Then, could you comment on the risk of silencing of MSH3 and inhibiting mismatched DNA repair pathway in neurons and dividing cells as astrocytes and microglia?
I mean, it's a high risk because it can cause, for many of the other genes in the mismatch repair pathway, they're associated with brain cancers. The brilliant thing about MSH3 is it's not associated with brain cancers. So if you can selectively silence the MSH3 gene in the brain, then potentially you won't have any side effects. So we have looked at some preliminary genomic instability. We've looked at other repeat loci in the mouse genes that have been known to increase with knockout of mismatch repair genes. And we don't see any increase at the 4-month time point. So we are gathering more data on that. We need to look at single nucleotide polymorphisms, I would say, a bit more, but we are quietly confident that silencing MSH3 should be okay.
So if I'm correct, I think mice don't have that many repeat expansions. So they have a relative short repeat expansions, and I think they don't have that many as we have in human beings. So are you also investigating this in other more human models?
We plan to do that. We've only looked at the mice just because that's what we've been working on. So we've had it easily available at the moment.
Okay, yeah, it might be nice to check this in the patient-derived cells. Yes, I think there is. Alex says thanks a lot for all the for the answers. That's also what Annemiek is saying. Thanks a lot for answering all the questions.
No worries.
Erin.
Yeah, great. So maybe we can just kind of finish off this session, Dan, with if you have any advice in how to get involved in patient advocacy. For example, your research trajectory started out as very like physical sciences and then moved into this type of work. And I know you've always been mindful of patient advocacy and Huntington's research throughout your career, but I'm wondering if you can maybe speak on kind of specific choices you made to help you end up in the position that you're in to be able to do what you're doing.
That's a great question. So I guess, to be honest, when I initially found out my mum had HD and that the effect it might have on me, at first I was like, I didn't want anything to do with the patient community. I was like, no, I just want to focus on, but I knew I wanted to help people. So I focused more on that. And then slowly but surely, I came to the realization of what a great community it is. I say that was through reaching out to local Huntington's disease associations, both in the UK and here and in Canada. I was in contact with those people, and I started there at like a low level, just meeting some people on a regular basis to talk about their experiences, and then started to take that further. There's Global Genes, which is a big rare disease place in the US that I would highly recommend you go to. On their website, they have a list for all the different rare diseases. If there are associations known, you can go on their website and they'll link you to the association or community. So it's really useful in that way. And yeah, I guess I just started just trying to meet some people, find a local association of whatever disease you're talking about and go there and chat to people and interact with them. Most of them are always willing to help.
Great. And I know also you've used a lot of your kind of science communication skills with your foundation to be able to provide kind of digestible information about what's going on in the community and the research to patients. And so how did you get involved in kind of making your own foundation?
Honestly, it was a bit of a, one day I throughout my PhD, I was always like, I didn't feel like I was contributing enough. You know, even though I was doing good research, you know, I was like, I really want to be impacting patients. And so one day I was just talking to my granddad. I was like, Granddad, I really want to start a charity. And he was like, funny enough, Dan, I was thinking about the same thing. And so I should highlight that from then on, he took the initiative and really led that. And so without him, that wouldn't be a thing. But it's doing some great work now. We donate, the things we donate for is, you know, wheelchairs. We give money per wheelchairs if people need that. We also give money if people say, hey, look, we want to go away on holiday with the patient, but because they need special support, they can't do that. They can't afford it. So we give money for those sorts of things, which I think is really important as well. And it's been a real pleasure, to be honest. Like every time we give enough donation, I'm proud of what we do. And I think from a communication point of view, I actually just started to do a bit of reading on science communication. I gave a Point of Science talk. I just started to try and do those things. At first, what I was doing was not great, you know, but eventually you find your way, you learn from other people, you speak to other people doing good science communication stuff. And that's how I ended up where I am today.
Wonderful. Well, thank you so much, Dan. This has been a really enlightening talk, great science, great advocacy, and we just want to thank you for giving us your time. And also, sorry, go ahead.
Thank you for the invitation.
Yeah, of course. You can come anytime, Dan. We also just want to remind the attendees and anyone who's watching at home, we have another webinar coming up scheduled for September 8th with Dr. John Watts, so that should be a good one. And then we'll have more. You can keep checking the website towards the end of the year. If you are interested in sharing your story, sharing your research, any science communication, patient advocacy, anything like that that you're working on, please get in touch with us. You can email info@oligotherapeutics.com or .org. And you can also reach out to us through Twitter.